Hi children, today I am going to take the topic Microsporogenesis. Microsporogenesis is the development of microspores from the microspore mother cells by meiosis. And remember, how many microspores are produced? 4 microspores are produced. So once again, what is microsporogenesis? The development of microspores from the microspore mother cells by meiosis. And this happens in the male reproductive organ of the flower, the stamen. So, a brief structure of the stamen. The stamen comprises two parts. The stalk, called filament, and the lobe called the anther. And remember, we have learnt in 11th standard, the lobe is usually bilobed. In angiosperms, the lobes are bilobed, like this, two lobes together. And not only that, in each lobe there are two thecas. So it is called dithecous. So the lobe is not single, it is bilobed with two theca in each lobe. Therefore it is referred to as dithecous. Now we'll take a cross section of the anther. When we take the cross section of an anther lobe, it appears almost like a butterfly. It is referred to as the tetragonal structure.
Initially, the anther is immature and young, and it comprises of 4 layers. The anther is surrounded by 4 layers. The outer layer called epidermis. See, the green color outline is called the epidermis. It is followed by endothecium, and then inner to the endothecium is the middle layer, and the innermost layer is the tapetum. So there are 4 layers that surrounds the anther, namely outer epidermis, the one indicated in green, followed by endothecium, which is pink in color, and then followed by the middle layer, white in color, and the innermost layer is the tapetum, which is indicated in blue color. The function of the first 3 layers, that is epidermis, endothecium and middle layer, is for protection. These layers are involved in protecting the anther and also helps in rupturing the anther lobe at the time of maturity to release the pollen grains. So 2 functions are provided by these 3 layers, that is protection and dehiscence. Dehiscence is the rupture of the anther lobe to release the pollen grains. And the innermost layer, tapetum, is involved in nourishing the pollen grains. The innermost layer is involved in nourishing the pollen grains. Now, inner to the tapetum, can you see a circle, pink circle? That indicates the sac called microsporangium, the pollen sac called microsporangium. In cross section, it appears circular, and there are 4 such microsporangia at the 4 corners of the cross section.
In an immature anther, the microsporangium comprises of compactly arranged homogeneous cells called sporogenous cells. See, the first microsporangium which I have indicated here, it comprises of homogeneous cells called sporogenous cells. So this is depicting an immature anther. Now as the anther matures or as the anther grows, there are changes occurring. So the second event is, the change is meiosis. For example, if this sporogenous cells, the number is 10, after undergoing meiosis, the number of microspores produced will be 40. That is 4 into 10, 40. So the next diagram is depicting the second stage where the sporogenous cells undergoes meiosis to form the microspores. So we have the second diagram within the same anther showing the microspore tetrads. See, the microspores are located huddled together to form the microspore tetrad. Next, again the anther is maturing. On further maturation, the microspores dissociates from each other. They separate from each other. So the next diagram is depicting the third stage where the microspores just separate from each other. And on further maturity, this is depicting the fourth stage. What happens? The microspores gets transformed into the pollen grain. The structure of a pollen grain. This is a pollen grain. It is circular in outline. It is surrounded by a layer called intine. Then outer to the intine, there's another layer called exine. And within the pollen grain, there are two cells, a vegetative cell and generative cell. The vegetative cell is very large and has an irregular shaped nucleus. And the generative cell is spindle shaped with a single nucleus. So once again, the structure of a pollen grain, the microspores get surrounded by two layers, inner intine and outer exine. And within the pollen grain, there are two cells. The large cell, which is indicated in blue color, is the vegetative cell. And the spindle-shaped green color cell is the generative cell. So we have reached the final stage where we have the full-fledged pollen grains. And now what happens? The layers surrounding the microsporangium ruptures. Releasing the pollen grains. All the pollen grains are released. So you should remember that each microsporangium, as I described, is circular in outline and is surrounded by four layers. Surrounded by four layers. If you see this diagram, which is the microsporangium? This is the microsporangium. The circular outline structure is the microsporangium. Like that, there are four microsporangium at the four corners of the anther. It is surrounded by four layers. From out to in, the four layers are outer epidermis, see the green one, outer epidermis, then followed by endothecium, then followed by the middle layer, and surrounding the microsporangium is the tapetum. So finally, when the anther ruptures, the outer three layers ruptures. It is the outer three layers that breaks open and releases the pollen grain. Then the role of the tapetum is to nourish the pollen grains. The pollen grains develop from the sporogenous cells. How did it develop? By getting nourishment from the innermost layer called the tapetum. Now all this was depicted in one tetragonal structure. The first stage, second stage, third and fourth stage. That is, as the anther matures, from a young anther as it matures, all this is depicted in one tetragonal structure. This should be depicted in four separate diagrams. So the four diagrams are these. So these four diagrams gives you a recap of the entire process of microsporogenesis. So the first event is the sporogenous cells within each microsporangium acts as the microspore mother cells. So in an immature anther, all the microsporangium comprises the sporogenous cells. So this is the first stage when the anther is immature. All the microsporangium, which is circular in outline, has a mass of cells called the sporogenous cells. And all these sporogenous cells serves as the microspore mother cells. Unlike megasporogenesis. In megasporogenesis, we saw only one cell acts as the megaspore mother cell. Here, all the sporogenous cells of the microsporangium acts as the microspore mother cell. Next, the second stage. Second stage is as the anther matures. What happens? The process of meiosis occurs. Meiosis occurs and the sporogenous cells gives rise to microspores. And the microspores occur as clusters. And these are called microspore tetrads. Again, on further maturity, third stage, only one event is occurring, dissociation. The microspores separate from one another. So you can see the third structure, the clustered microspores detach from each other and they are seen separated. And final stage, what happens is the microspores gets converted into a fully fledged pollen grain. This is a full-fledged pollen grain. See, it is circular in outline, surrounded by two layers, outer exine, inner intine. The outer exine is made up of a substance called sporopollenin, and the inner layer called intine is made up of cellulose and pectin. Now within the cell, there are two cells. It is not single cell, it is double celled. Vegetative cell, yellow is the vegetative cell which is large in size and with an irregular shaped nucleus. And at the base is a generative cell. Look at the shape of the generative cell. It is spindle shaped with a prominent nucleus. This is a pollen grain. So this is what happens in the fourth event where all the microspores becomes the pollen grains. So we have all the microsporangium with the pollen grains fully developed. Now it's time for rupture. Which are the layers that are going to be ruptured? If I enlarge this diagram to show that. So the layers that are going to be ruptured are the green, which is green depicting epidermis, endothelium, middle layer and tapetum. They rupture and the pollen grains are released. Similarly, it occurs in all the sides. Pollen grains are released. I hope you have understood today's topic.